Okay, let's see. I'm going to run through that to-do list once again real quick, and then we're going to get started. I feel like we'll start with our diagrams of skeletal muscle tissue. But just to remind everybody with where we're going, we just got done with exam 2. So you've got exam 2 and practical 2 passed back. We should be able to get through muscle tissue today. The first PowerPoint, we compare skeletal muscle tissue, cardiac muscle tissue, and smooth muscle tissue. You can see I've got some models on the side of the room where we'll look at what cardiac muscle tissue and skeletal muscle tissue look like compared to smooth muscle tissue. We'll compare the three basically. We'll focus mostly on skeletal muscle tissue because you are in voluntary control of this type of muscle tissue. So if you want to cause a contraction, you can cause this to contract or relax. And at the end of this, we'll talk about the neuromuscular junction. Maybe this is the physiology part of the discussion where we talk about how these muscles are fired, what causes these muscles to contract or to relax. And this might be worth taking a snapshot of real quick. We talk about this a lot. We talk about it for the rest of this semester and even next semester when we talk about cardiac physiology. Cardiac muscle, again, any type of muscle tissue isn't going to be able to contract or relax without some type of electrical event that precedes it. So I tried to underline these terms that we will use to describe these electrical events like depolarization and repolarization. Those describe electrical events that always precede one of these mechanical events like contraction or relaxation. In fact, what's I think neat about this lecture is that we start to talk about neurons at the very end of this lecture, and then we spend the rest of the semester talking about neurons. Neurons, we know, can send a short electrical signal, and if you've ever done any kind of muscle rehab or anything like that where you've put one of those electric stim machines up to a muscle, you can just hit it with an electric shock. Or don't do this, we can just do the thought experiment. If you took a paper clip or something like that and stuck it into one of these sockets, you would feel electrical currents that would cause some muscles to contract. So muscles can respond to electrical events and it causes them to contract or relax. Neurons are going to send those electrical events. All of these muscle types are going to respond to electrical events. We'll start with muscle tissue. So again, we're going to look at how muscles contract and relax, and then on Thursday we kind of zoom out just to list muscles of the body. And on the lab list you've got the front page of it takes us through things we need to know on these muscle tissue models. And if you want to see where these muscle tissue models are located, I tried to scatter them on this side of the room. So next to models of skeletal muscles, we have these skeletal muscle tissue models. We're going to draw these skeletal muscle cells. This whole thing is one cell. It is highly modified. We're going to compare these skeletal muscle cells to cardiac muscle cells. Let me get the bone out of the way now. Cardiac muscle cells have some similarities, but they do have some things that are unique, like this branching nature to them. And we talked about those intercalated discs, which are unique to cardiac muscle tissue. You only find it in the heart, so we've got the heart wall as a large-scale representative of cardiac muscle tissue. And then smooth muscle tissue, you've got these spindle-shaped cells that don't have the striations, these alternating light and dark bands like we see with skeletal and cardiac muscle tissue. You find this smooth muscle in the walls of blood vessels in the digestive tract. So we've got some examples of where you find it. Quickly, while I'm thinking about that, I'm going to come back here and grab this one because we're certain to see this model later on in the second half of the semester. This is a cross-section through the digestive tract. And we can see two thick layers of smooth muscle there. So let's set that next to the smooth muscle tissue model. Everything else on that lab list takes us through muscles of the body. If I get a chance today, then maybe we can preview those. If not, we should be able to tackle all of that on Thursday. It's only 2 lectures that's part of the muscle unit, or unit number 3, and I think 75% of the slides are set up to try and for us to get through them today. We'll see how far we can get through. But with that out of the way, any questions before we jump into this at all? All right. We were drawing a long bone last time where we'll just label this the bone. We had our typical long bone, and kind of attached to this long bone For the first time, we're going to draw this bone to muscle connection that we're going to label a tendon. Let me get a different color here. This represents our tendon. We know a tendon is a muscle to bone connection. And here's where we're about to sketch what we see when we're looking at this muscle model. You're looking at what we would describe as a muscle belly. The outside surface of this muscle is going to be covered in a layer of connective tissue. Let me try and expand this thing out. Cut off a little bit of that. So this thing that I'm trying to draw is the muscle belly. It's the very outside of the muscle. Again, when you touch the surface of one of those muscle models, you're touching the muscle belly. And the muscle belly is going to have just on the outside of it a layer of protective connective tissue. I'm trying to put it in this orange color. It's connective tissue. It's just to the outside of the muscle belly. So a good term that describes this type of connective tissue is going to be the epimysium. So epimysium is just to the outside of this muscle belly. It's a type of dense connective tissue. So is a tendon. It's the same type of dense connective tissue. Those collagen fibers twist together, kind of like the end of a Tootsie Roll that twists up. So this kind of seals together the tendon and the muscle belly and attaches it to that bone. So we've got our epimysium to the outside. If we look inside a muscle belly, what we would see are these kind of irregular shapes. Let's pull one of these out. So sometimes it's good to have an eraser if you're doing it in pencil. But I'm just going to label each one of these things with an F. This is going to be what we call a muscle fascicle. Let me see if I can squeeze the definition of muscle fascicle in here. A muscle fascicle is going to be a bundle of muscle fibers. A bundle of muscle fibers. So each one of these, I've just put an F in the middle of those, but if we look in the middle of each one of these fascicles, what we would find are these large circular structures. Each one of those is a muscle fiber, and I'm going to pull one of them out. So that thing is a muscle fiber. We doing good so far? I feel like there's one thing, let me finish this thought real quick. A muscle fiber, these really long cylindrical shaped cells are large enough that you can see them with the naked eye. And I described something about like ripping apart brisket or pulled pork or fajita meat. When you see those strands of meat, that's an actual cell that you can see with the naked eye. It's so large that it was originally mislabeled. It was just called a muscle fiber. But what we know now with microscopes, that a muscle fiber is the same thing as a muscle cell. That whole thing is one cell. And when we look inside the cell, you're going to see other organelles. For example, since this is one muscle cell, we're going to see organelles that are part of this muscle cell, like a nucleus. I'm just going to label this as a nucleus. We know skeletal muscle cells have multiple nuclei in them. They're going to have other organelles too when we look inside there. But let me back up just a second. We have to the outside of the muscle belly this connective tissue called epimysium. If we look to the outside of the muscle fascicles, there is also a layer of connective tissue that surrounds the muscle fascicles. It's going to be called perimysium. We would see this perimysium to the outside of all of these fascicles. So just for time's sake, I'm going to fill in this paramycium to the outside of all of these muscle fascicles. All right, and if we look inside one of these muscle fascicles, we have our muscle fiber. And I'll just use this color to show that to the outside of each muscle fiber, there is also a layer of connective tissue surrounding each individual muscle cell, each muscle fiber. This is called the endomysium. So you would find it to the outside of each one of these muscle fibers. You just squeeze it in there for time's sake. Okay, I'm just trying to set down these two skeletal muscles, which is what we're trying to sketch. This whole model is one muscle cell or muscle fiber. When you flip this thing around, all of these collagen fibers, this little piece of connective tissue is the endomysium. And when we look inside, all of these structures, like these long cylinders, these are organelles. This net-like structure, it's an important organelle of skeletal muscle that houses calcium ions. Later on, when we talk about a muscle actually contracting, it requires calcium ions to contract. And if we want the muscle to relax, we need to get rid of calcium ions. Here's where those calcium ions are stored, and when we want calcium ions to move out of the fluid of the muscle cell, they have to go back into this net-like organelle that we're going to call the sarcoplasmic reticulum. You can see other things in here in blue, like these T-tubules. So there's lots of things inside the muscle cell that we need to actually see. You've got a couple of models of that. This one, they're using a blue color to show T-tubules and kind of a tan color to show this sarcoplasmic reticulum. The other model is showing the T-tubules in kind of a red color and the sarcoplasmic reticulum in kind of a white color. This is also a skeletal muscle fiber, the same thing that we're trying to draw. And I like it. It's a little bit more zoomed out than the other two skeletal muscle fiber models. But what we're looking at here is an actual muscle cell, same thing as a muscle fiber, and you can see this endomysium just to the outside. Okay, so now I found the model that I wanted to show. Now let's take this a little bit further and start labeling the actual muscle cell, the muscle fiber. And can you see just kind of protruding through the surface of this, those multiple nuclei that are part of skeletal muscle cells? I put a few of them here in blue. But there's some other structures that we can see, like these rod-like structures that fill each muscle cell. I'm going to put just a few of those things in there. I'll just keep using the red color. This muscle cell is going to be full of these rod-like organelles, and I'm just going to pull one of them out. I know I don't have a lot of room here. It's getting relatively small, but this little structure is an organelle of the muscle cell that we're going to call a myofibril. This myofibril is actually going to be made out of 5 different types of proteins. Bless you. And in a second, we're going to erase this entire diagram, and we're going to zoom into this myofibril. So just a heads up of where we're going. The myofibril, it's I sometimes think of this myofibril as like a long chain. Just like a chain is broken up into individual links, this myofibril is going to be broken up into individual links as well. Each link, just real quick to point to one of those individual links, that link is a sarcomere. These sarcomeres are sometimes thought of as the functional unit of a muscle cell because it's each sarcomere that's going to contract. The myofibril is the contractile organelle of this entire cell. And when we're thinking of organelles, there are other organelles that you find inside a cell. I'm just going to put as little orange dots here. These are going to be organelles that I'll label as mitochondria. So this is actually a typical cell. Typical animal cell, we can see a nucleus, we can see endoplasmic reticulum, we can see mitochondria in there. Skeletal muscle cells are highly modified where you almost don't see any of the fluid that you see in a normal cell. This fluid of a normal cell we call cytoplasm. You see all of these organelles floating around in the cytoplasm of this cell. Muscle cells were misnamed to begin with. They were so large, again, they were originally called muscle fibers rather than an actual muscle cell. So the rest of some of these components of a muscle cell also get kind of a weird name. For example, the surface of this muscle cell, what we would normally call, I'm just going to draw this out here with a black line, what we would normally consider the plasma membrane. The plasma membrane of a normal cell, when we're talking about muscle cells, it's called the sarcolemma. So sarcolemma and plasma membrane of a muscle cell are the same thing. And I just talked about that cytoplasm of a normal cell. Let me try and squeeze this out over here. If we looked inside a normal cell, what the fluid would normally be referred to as cytoplasm. But in a muscle cell, everything gets a different name. Instead of calling it cytoplasm, we refer to the fluid inside of a muscle cell as sarcoplasm. The main organelle floating around in that sarcoplasm is this myofibril. That's the organelle that actually contracts. And to see what's going on with how the organelle contracts, we're going to need to zoom in closer to this muscle cell and those myofibrils. So I'm tempted at this point to pull some of those slides down and kind of catch up in the PowerPoint slides with skeletal muscle tissue. We'll compare it to cardiac muscle tissue and smooth muscle tissue. But then we're going to come back and draw this muscle fiber and see exactly how those sarcomeres start to contract and what are those fibers that build this myofibril. There are 5 that we will list.
Can you explain the sarcomere?
Sarcomere, yeah, this is going to be, like if you think of a long chain, each link in that chain would be a sarcomere. In my chain analogy, this myofibril is the chain, and each link in that chain is a sarcomere. And we're going to draw a sarcomere in a second. You're going to see that a sarcomere, it would be like a link in the chain that can just like become half as big. It contracts and becomes half as big as it normally is. So you can imagine a long chain where each link in that chain would contract. It's a way for the whole chain to become like a half or a third of its original length. So muscle cells contract by each individual sarcomere, each functional unit contracting, which it might help if we can see some actual slides. So I'll leave that up for just a second and get these slides going. Is anybody a pro at this? All right, it's getting tied off. Okay, hopefully that's a little bit better. If you follow along with that class handout, we've already answered a few of those things from the first page. But let's compare when we look at muscle tissue. We've seen these slides before. We've seen skeletal muscle tissue, cardiac muscle tissue, and smooth muscle tissue. We know here we're looking at skeletal muscle tissue. You can see these long fiber-like cells. Originally called muscle fibers, we know these are individual muscle cells. When we look close at each muscle cell, you can see those alternating light and dark bands. We're about to see where those come from. But you can see all the darker little dots, those are individual nuclei. So each cell is multinucleated. We compared this skeletal muscle tissue to cardiac muscle tissue where you can still see those alternating light and dark bands just faintly. But unlike skeletal muscle tissue where all of these cells were kind of long cylinders, in cardiac muscle we see some of those cells branch and fork. In addition to this branching, the other thing that we notice are those intercalated discs. Those are unique to this cardiac muscle tissue. And then we have smooth muscle tissue that doesn't look like any of the other two types of muscle tissue. We have these long spindle-shaped cells. They kind of taper at either end. They have a single nucleus in the middle. They don't have this striped or striated appearance. We talked about places where you find them lining the digestive tract or lining blood vessels. We spend most of our time talking about our skeletal muscle tissue. So we've already drawn on the board one of these long fiber-like cells with multiple nuclei. And where we're about to go is inside that cell to see some of the organelles that make up this skeletal muscle cell. And the one that we're going to look at, those myofibrils, are going to be responsible for those alternating light and dark bands. Okay. General characteristics of all muscle types, whether it's skeletal muscle tissue, cardiac muscle tissue, or smooth muscle tissue. All muscle tissue are going to have these four characteristics. They have this excitability. That just means that they can respond to an electrical current. They're excitable. You hit a muscle with an electrical current, it's going to cause that muscle to contract. The second thing that I just said, this contractility means that muscles can shorten whenever they're hit with an electrical stimulus. This contractibility, shortening, what pulls around on some of these bones. Extensibility, that means you can take a muscle that is not contracted and actually stretch it out longer than it normally would be at rest. So muscles are stretchable. They have this extensibility. Now, the other thing, and we're going to draw this in a second to see where these last two characteristics come from, this extensibility, the fact that you can stretch a muscle out longer than it would normally be at rest, and then when you let that muscle go, it goes back to its original shape. That ability to recoil is what we call this elasticity. All right, here's a picture of a muscle cell. This is an embryonic muscle cell that's starting to develop, but you can still see this excitability. When it's hit with an electrical current, it has this ability to contract. Okay, normal muscle functions are going to include movement of the bones, movement of body fluids. Like I mentioned, the smooth muscles and the walls of the blood vessels. Muscles are also there in addition to causing body movements. Muscles are also there to help keep the body stable. So we'll talk about these stabilization muscles that maintain body posture. I feel like this is somewhere in the lecture, but you see this in watching, like, sometimes it happens in sports where there will be these collisions, most often in football, where players will have these head-on-head collisions, and you'll see if muscle tissue, like if there's a signal that is stopped between the brain and skeletal muscles, you'll see these muscles that are constantly tense to maintain body posture, they will go limp, and you'll see somebody just look like a rag doll hit the ground, you know. Anyway, muscles are for movement and also just for maintaining stability and maintaining posture. Muscles, in talking about maintaining stability, muscles around a joint are the main things that give the joint stability. So if you are rehabbing a damaged joint, it's typically all about doing exercises that build the muscles around that joint. And then the final thing, we can say muscles are able to generate heat when it gets too cold. We're not having to worry about that right now this time of year, but again, walking into a cold room or remembering how it was in wintertime, if you get too cold, your muscles can involuntarily shiver, and that generates a little bit of body heat. Whoa. We jumped over this picture, was just a cross-section through a muscle belly, kind of like the one that I tried to do here, but I didn't try and draw any blood vessels in a muscle belly. And this shows us that if we were to actually open up the center of a muscle belly, there are these major arteries that run through the middle of each muscle belly, and they deliver nutrients to each of those muscle fascicles. We can see just to the outside of the muscle belly, there is the epimysium. We can see surrounding individual fascicles is the perimysium. And looking inside one of those fascicles, each one of those structures is a muscle fiber, what we know as an actual muscle cell. Other general terms to introduce real quick. When we talk about muscles of the body, it will be the case that some of them are going to be referred to as being indirectly attached to bones versus this more direct attachment. A direct attachment is when these muscle fascicles actually anchor to the bone itself. That periosteum that grows around the outside of bones, those muscle fibers, if you have muscle fibers that attach directly to periosteum, it's a direct muscle attachment, as opposed to something like this gastrocnemius muscle. We can see there's a long Achilles tendon where there isn't any direct muscle fascicles that connect to the bone. Instead, those muscle fascicles connect to a tendon. So what I was attempting to draw here is an indirect attachment between the muscle and the bone. There's a tendon in between. Okay, so now to naming the structures that we see when we open up a skeletal muscle. We've already sketched some of this, so I can catch up with the slides. We know epimysium surrounds the outside of a muscle. Paramysium is just going to surround those muscle fascicles, a group of muscle fibers. And endomysium surrounds individual muscle cells. We've gotten this far in our diagram where we have a tendon that has a muscle belly coming off of it. We've opened up that muscle belly to see all of these individual fascicles. Each fascicle is surrounded by its paramysium. We've looked into one of those fascicles and pulled out a muscle fiber. We know a muscle fiber is the same thing as a muscle cell, and we can see organelles of a typical cell like those nuclei. We know that the fluid inside here is called sarcoplasm. There are other organelles like mitochondria that are stuffed in between those other little cylinder-shaped structures. Those are the organelles that we call myofibrils. They completely fill these muscle cells. I guess my brain is thinking of a couple of side notes, and maybe one thing that's interesting, since we're looking at a muscle cell real quick, if you ever wonder like what grows whenever you work out. Like if I wanted to dedicate the rest of the summer to eating right and working out and getting my muscles larger, what would happen is I wouldn't get any more muscle cells. I would have the same number of muscle cells as I do now as I would at the end of the summer. But what grows, what changes is the number of myofibrils within a muscle cell. Remember, cells are constantly recycling their organelles, and if it's constantly a case of use it or lose it. If you use your muscles a lot, then this cell is going to go, man, we need to manufacture more of these myofibrils so that we can get stronger. And if you're not using those muscle cells, then these myofibrils will be recycled. You'll have fewer myofibrils, and those muscle cells won't be as strong. Okay, so number of muscle cells don't change, but the organelles inside those muscles can fluctuate. Okay, this again is just kind of a snapshot of what we were trying to draw on the board. I wanted to take a snapshot of the outside of the muscle so that we can see epimysium to the outside of the muscle belly. Paramysium is around each individual fascicles. And then right behind the screen here, we've pulled out one muscle fiber, and we've listed some things inside there like the nuclei, the fact that the plasma membrane is called the sarcolemma. And we mentioned those things like the mitochondria that we can barely see in there. The most notable organelle is this contractile organelle, that myofibril. And I was mentioning how this thing is like a chain that's broken up into individual links. And I don't know if you can start to see those individual links on this chain that we call a myofibril yet. But if I can zoom into this one picture, here we are. We're zooming in a little bit further. We can see that contractile organelle, this myofibril. And you can almost see how it's broken up into those individual links. We said this is the dominant organelle of muscle cells. It takes up over 80% of the volume of cells. And here's where I also want to spend 15 minutes getting off on a side note, which I'm going to try and just spend 5. But there's something interesting about just the anatomy of a muscle cell. If I can zoom into that just a little bit bigger, we've zoomed into this muscle cell. And I want to point out that these myofibrils, like we said, they take up most of the space in a muscle cell. There's very little empty space. You can see organelles like the nucleus and even these mitochondria are squeezed in between those myofibrils. What is also inside these muscle cells, well, I hate to erase that, or even that. This is where I wish I had more board space. Is it okay if I erase this for a second? I'm gonna take like 5 minutes and do this. Sorry. I think this is important, I guess, because I always try and relate, real life situations to what we're learning. And this is a common one, I guess. I always share my own family issues. My dad is a terrible diabetic. Odds are, genetically, I'm likely predisposed to being diabetic as well. And what does that mean? And how is that going to affect us later on? Here is a normal body cell. And at some point, this is going to come back in the class. So it's extra information for now, but at some point, we're going to draw this normal body cell. Normal body cells, their favorite fuel is going to be glucose. Let's grab a couple of extra colors here. A glucose molecule is too big to just move through this plasma membrane. Label this our plasma membrane. And label this as our glucose. Glucose has to somehow get into this cell if the cell is going to break it down and use it for an energy source. I mentioned glucose is too big to cross this plasma membrane on its own, but normal body cells, they have locked away in the cytoplasm of a cell. They have vesicles. This is a vesicle that contains, there's all kind of different vesicles inside a cell that are filled with a variety of different things. But one particular vessel, one particular vesicle contains a protein. I can't figure out what color I want to draw it in. I'll just draw it in a blue color. This I'll just draw as a box. But that protein is a GLUT. To be specific, it's a GLUT4 protein. GLUT just stands for glucose. T is transporter. 4 just means it's the 4th one that was discovered. There's 20 different glucose transporters. We'll talk about most of them at some point. GLUT4 is a glucose transporter that is currently inactive. And you find these in all body cells. Their job is to allow glucose to get into the cell, but they typically don't work because they're not attached to the plasma membrane. The only way that you can activate these GLUT4s is if there is a receptor on the surface of this cell that is hit with insulin. So insulin is this chemical signal that's going to come from the bloodstream. When it docks on this surface receptor, it's going to kick off a cascade of reactions. I'm simplifying a series of events here that basically lead to the activation of this GLUT4. So insulin leads to a cascade of events that causes this GLUT4 protein to actually come over here, and instead of being just kind of floating around in the cytoplasm of a cell, that GLUT4 gets activated, attached to the plasma membrane. Now in its active form, it's going to allow glucose to get into the cell. So now this cell with its active glucose transporter is able to feed on this glucose, and it's quite happy. Normal body cells, they require that you have those insulin receptors that work, right? This assumes several things. This assumes that your body is producing insulin. It assumes that the receptors are okay on the surface of those cells, that they're docking insulin. And then once they dock insulin, this whole cascade of proteins on the inside goes correctly and actually activates this GLUT4 protein. All of these things are going to get glucose levels to come down in the bloodstream. And in fact, there are different medications. I mentioned my dad. He has a brother and a sister that are both type 2 diabetics, and they're on different medications because different stages of this process can be faulty, and different medications will affect different steps, right? Some affect how sensitive this receptor is to insulin. Others increase the amount of insulin or decrease the amount of insulin. But what's interesting, to get back to the point here, skeletal muscle fibers also have those GLUT4 proteins. But what's unique about the anatomy of this, there's just not much available cytoplasm in skeletal muscle cells. So if you contract these cells, which we said all of them contract when you activate, when you work out or walk around or use these muscles, by contracting these muscles, what it does is it squeezes all of these organelles, like the mitochondria, the nucleus, out to the very edge of the cell. It even squeezes these vesicles that contain GLUT4 proteins. I guess this is my long-winded way of saying when people have elevated blood glucose levels, one of the things they say is just exercise is one thing that can bring down blood glucose levels. And the reason that activating skeletal muscles can bring down glucose levels is because you can physically activate GLUT4 proteins. It's the only cell in the body in which you can physically activate GLUT4 proteins. Normally, the only way to activate them is through this cascade of chemical signals, medications. Skeletal muscles, that's not the case. Anyway, I'll peel away from any other side things that are popping into my head. I'll leave that up for a second, but I just wanted to make that point. Since we're talking about anatomy and the anatomy of these skeletal muscle cells, there's not a lot of available cytoplasm. So when these cells contract, all organelles get squeezed, pushed out to the very perimeter of these cells, and that happens to be helpful for GLUT4. Let's talk about the other thing that's labeled on here. Now that we're zoomed in this far to a muscle cell, you can see that myofibril. How I was talking about it's like a chain, and each link in that chain is a sarcomere. We're about to look at those. The other thing that you can see now that we're at a bird's eye view of this myofibril, you can see how the collection of these myofibrils are making these alternating light and dark bands. There's an easy way to remember dark bands versus light bands. The dark bands are always referred to as A bands. Light bands are always referred to as I bands. And one way that you can remember that is that you need the I in order to spell light. So I bands are light bands. There is no I in dark, so you wouldn't confuse an I band for a dark band. There is the letter A in dark, and dark bands are considered A bands. So maybe think of that second letter in the word as telling you whether it's a dark band, A band, or a light band, which is an I band. So these alternating A and I bands form these striations. So we're going to have to zoom into that. Let me see if I mentioned anything else in our zoomed out version of this muscle cell. So the most common organelle of this muscle cell are those myofibrils. So this is where we can go back to our diagram here and see if we can draw one of these myofibrils. We're going to see that it's broken up into these individual sarcomeres. And I guess just one more thing before I erase this diagram and start to draw our myofibril. You see that I snuck another picture in right behind this. And that is a picture of what we consider these slow twitch muscle fibers or these fast twitch muscle fibers. This is a cross-section through a skeletal muscle cell. Each one of those is a muscle fiber or a muscle cell. Some of these muscle cells are light staining, while other muscle cells are rather dark staining, and other ones are kind of this intermediate between dark staining and light staining. The reason that there's a different amount of stain that's being picked up by these muscle cells, it's an indication of how many mitochondria are there. So if we go back here, these mitochondria are squeezed in between these myofibrils, and these cells are full of mitochondria. So they take on a dark stain. These have little to no mitochondria in them whatsoever. We'll see this at the very end of the PowerPoint too. I just wanted to I threw this slide in here early because just prior to this slide, we saw those mitochondria that are nestled in between these myofibrils. And it's really the number of mitochondria that give these muscle cells their dark appearance, or lack of mitochondria give them their light appearance. I'll come back to that in just a second. The ones that don't have any mitochondria are considered the fast twitch muscle fibers. The ones that are loaded full of mitochondria are the slow twitch muscle fibers. The ones that are in between are intermediate. Okay. So just right now we're thinking of muscle fiber, whether it's fast or slow, that's really talking about how many mitochondria are there. But before we focus on mitochondria, let's get back to this myofibril, this organelle that really causes the contraction of a muscle cell. So You know what? I'm going to leave that up. I'm just going to see if I can squeeze it in on this side. Okay, so we've got one, in fact, that rod-like structure that's coming across there, that is the myofibril. And I know the screen behind me here, we were drawing a muscle fiber. I had a few of those rod-like myofibrils coming out. What we're going to do on this side, I'm going to put, once again, this myofibril. But we're going to draw it a lot larger. Let's make this myofibril just stretch all the way across the surface. I've got at least that much room. Now this myofibril we know is going to be broken up into individual links, so let me get a different color. And I'm just going to put this zigzag structure down through this zigzag structure is what we're going to call a Z-line. So here's our other Z-line. Between the two Z-lines, this is a sarcomere. So we can see those little blue squiggly lines. Those are two Z-lines, and in between those two Z-lines, we've got one sarcomere. That's our functional unit. In the middle of that sarcomere, you can see three of those lines. I'm going to get a purple color here to put three of these lines right in the middle of this sarcomere. The collection of three lines in the middle of the sarcomere is called the M line. M line is in the middle. Maybe you can remember M line because it's in the middle of the sarcomere. It's also easy to remember because attached to this M line, we're going to see there is the myofilament that we're going to call myosin. Come to think of it, I am going to have to erase this thing over here just because I want to put us a list of things that we're about to write. Is it okay if I erase this? Everybody's good? Okay, let's just put a little a legend of what we're going to try and fill out up there. A myofibril let's review. In fact, let's do it like this. If we're thinking of let's just put in red muscle tissue. I'm going to start with muscle belly. We know that this muscle, if we look inside a muscle belly, there is a muscle fascicle. If we look inside a muscle fascicle, we said muscle fascicles are going to contain a bunch of muscle fibers. We know muscle fibers that's the same thing as a muscle cell. Just a muscle cell. If we look inside the muscle cell, that's when we find organelles like this myofibril. This is the contractile organelle. So our contractile organelle, this myofibril. But if we look inside this myofibril, we're going to find myofilaments. In fact, there are 5 myofilaments. Hopefully I'm not cut off there. We're going to list these myofilaments. In fact, I'll just do that right now. I'll try and stay color coordinated when I do. The first myofilament that we're going to list is called myosin. The other one that we're going to list is called actin. Then we have a few others to list. There is a protein that we call titin. Where's my green colors? I know, but there's 2 shades of green that I'm looking for. That's what's throwing me off. So instead of using green, let's, I don't know how distinct these two shades of red are, but these are going to be the other two myofilaments. I'm going to make the darker one what we call troponin. And then the more pinkish one, what we call tropomyosin. I like those being in a similar color because they're always right next to one another. To use my Charlie Brown analogy, there's a character in Charlie Brown, one of them, and I forget their names, but one of them drags around like a security blanket everywhere they go, right? And this is that character. Troponin is that character, that Charlie Brown character that always carries a blanket around with him. And tropomyosin is that blanket. So we're going to see wherever we see troponin, it's going to be dragging this tropomyosin with it.
Did you say the myofibril, it's called a contractile organelle? Is it because it contracts the muscles that way?
That's right. That's going to be the organelle that's responsible for the shortening.
Thank you.
No problem. In fact, we're kind of getting into it here. Let me kill that for a second. Unhog tie this. Let me finish this thought now that we've got a little bit more space. So that's right, this is the myofibril that we're attempting to draw. We can see how it's broken up into those individual sarcomeres. So now that we've got a little bit more space, let's expand that. If we expanded it even further, we would see, we can put another Z line in here. And between two Z lines, this is another sarcomere. So sarcomeres are these repeating segments. In the middle of each sarcomere, we have this M line. The M line is going to have myosin filaments attached to it. So that's basically where we left off. We're talking about these myofilaments, these 5 myofilaments that make up a myofibril. Like if we look inside of a myofibril, this is the organelle that contracts. We're going to see these different myofilaments. I wanted to have a little bit more space to describe these. This tropomyosin that I described, this is going to be a long thread-like protein. So long thread-like protein. Troponin, this is going to be a rounded protein. We'll see, in fact, you have sketches of these on the back of your class handout for today. The rounded protein is troponin. The long thread-like protein is tropomyosin. Titin, this is a spring-like protein. What can we say about actin? Actin, this is going to be the main thin filament. And what's unique about this actin, let's add this. Actin is going to have an active site. What's unique about this active site is this active site is what myosin bonds to. Let's say it this way. Myosin bonds here. Bonds to that active site. If it's available, myosin will try and bond to that active site. But we will learn myosin is usually prevented from having access to this active site. It's blocked away. Myosin is the main thick filament. It's the first filament that we have up there. What's unique about myosin is myosin is going to have these myosin heads. I'm going to add these myosin heads to our sketch. And notice when I'm putting these myosin heads on there, the myosin heads are pivoting away from this M line. So over here, let's put myosin heads that pivot away from the M line. And since we have another sarcomere right here, I'm just going to load this one up with went the wrong way there. Myosin heads. We doing good so far? We got our M line, we got our Z line, we got our myosin in there. Let's add the next one, which is actin. I'll stay color coordinated. The actin is actually going to come off of this Z line. And actin is going to reach in between those myosin heads. It doesn't touch the M line. It gets close. We'll put some in the opposite direction. We even have some Z lines over here. So I'll put these thin actin filaments. They're always attached to a Z line. And they reach off in either direction from a Z line. Just to list this, we've got actin. We've got myosin. The only other myofibril that we're going to need to draw is this titin protein. Titin is another one of these thin filaments. It's not the main thin filament. In fact, when we're thinking of these myofilaments, there's only one thick myofilament, and there are several thin myofilaments. Actin is the main thin myofilament. Titin is another one of these thin myofilaments, and all of the thin myofilaments are anchored to this Z line. The titins, they come off of this Z line, and they attach to the end of the myosin. So the ends of the myosins aren't just flopping around. Each one of them is anchored to the Z line by way of this thin titin protein. So let's anchor each of those floppy ends of this myosin with a titin protein. Even we've got one over here. Let's finish that out. So I've got just about everything that we need to add on here. We've got our spring-like titin protein. We've got our actin and our myosin. The only ones that we don't have to draw on there, but we do need to know how they operate, are troponin and tropomyosin. Before I describe the actions of those real quick, let me add one thing on here. Jeez. Before I get those mixed up, let's grab this brown color. And I'm just going to find the ends of these myosin filaments, the ends of these purple filaments, and I'm just going to go right down this board, and I'm going to put a dotted line that kind of skirts the edge of those thick myosin filaments all the way down. I'll do the same thing on this edge. I will expand my titin protein over just a little bit there. We can even do the same thing over here. Since I have another sarcomere, I'm going to put dotted lines that skirt the edge of the thick myosin filaments on either side. Now that I've done that, if we step back over here, you can see that these parts of the diagram in which there are myosin filaments, anytime where there are thick filaments present, this is going to pick up a lot of stain, and it's going to stain dark. So this is going to be what we call an A band. A dark band. And we know that A bands are flanked on either side by what we call I bands. And if we look at what you find in an I bands are going to be made up of thin filaments only. There are no thick filaments in an I band. We've got the Z line, we've got actin and titin. Same thing over here. I could have put in more titin spring-like proteins coming off of this side. I bands are light staining because they only have thin filaments. A bands are dark staining because that's where you find the thick myosin filaments. If I'm counting number of points on that, we should probably be able to get at least 6 or 8 points off of that sketch, because what we're diagramming here is that main contractile organelle of a muscle cell. And we can see how this contractile organelle is broken up into links, like a chain. And each link is called a sarcomere, and it's each sarcomere that's going to contract. I realize we've just set up the anatomy to this yet. Now we need to start talking about how they contract. What is it that determines whether these muscle fibers are going to shorten or not?
On the lecture handout, it says M line and Z discs. Is there a difference between Z disc and
Z disc and Z line, either one of those terms is right.
Thank you.
No problem. I just want to add this one little thing to it real quick before I erase this. Remember that just to the outside of the muscle belly, if I were to put like a corresponding layer of connective tissue, we would see corresponding to the muscle belly, there would be the epimysium. And then if we look inside that epimysium surrounding the muscle fascicle, we said there was the paramysium. And if we looked through that paramysium surrounding individual muscle cells, there was the endomysium. And if we go inside an actual muscle cell, we're now inside a cell. So the fluid that we would typically call cytoplasm gets given a different name. We call it sarcoplasm. And now that we're floating in this sarcoplasm of a cell, we can see organelles like the myofibril, the mitochondria, the nuclei, all of those other organelles that we typically see. The main organelle that we see are these myofibrils. And again, we're about to talk about these myofilaments. We've got 3 of them listed on the board. We just need to talk about this troponin and tropomyosin. I'm going to need to pull some slides back down. Is everybody good with that so far?
I'm sorry, I don't mean to interrupt you.
You're good.
On the lecture handout, can you go over number 6 to make sure that I told you?
What does that say real quick?
It's saying one sarcomere extends between 2, and I put Z discs.
That's right. Z discs or Z line. That's right.
And then myofibril actin overlaps the myosin in the that is the I band.
So actin will overlap myosin. That's going to be in a dark band. Anywhere there's myosin, anywhere there's that thick filament.
Well, this one says actin. Actin overlaps myosin in the, you said I band?
Yep. Here's the diagram I think that you're thinking about as soon as this thing pulls back up. So we went away from the slides when I just wanted to outline the myofilaments that you can see in this picture. So we've listed these Z discs or Z lines. The blue here indicates the actin filaments. The red is obviously the myosin. You can't see the little tightened spring-like protein in there. There's a lot of words in this. I'll just read through these to make sure that we've listed everything. We talked about the thick filaments. Those thick filaments run the entire length of the A band. I know I've just covered that up, but we could list this thing right here as an A band. Anywhere there are thick filaments is where we find an A band. Thin filaments, we said they attach to the Z line. If we're looking at an I band, those are only thin filaments. Z disc and Z line are the same thing. I talked about the M line, which is the middle of the sarcomere where all of those myosin filaments are anchored to. But what I didn't draw on here because I didn't want to overload this thing with lines, there is what's called an H zone. The H zone is just this part of the A band where there are thick filaments only. You were just talking about a place where there's thick and thin filaments. So that's where I thought you were thinking about this type of picture. So there are places where there are thick and thin filaments. This would still be a dark staining area, one of those A bands. But there is a middle area here where there are thick filaments only, none of the thin filaments.
And that's the myosin, is the thick.
Myosin is the thick filament. And there's a part right in the middle of a relaxed centromere where there are no thin filaments. It's just thick only. That's the H zone. There it is right there, H zone. So here's where it's kind of zoomed in. We see the labeled I band kind of flanking either side of this A band. I think we've got everything labeled on there. You can see the little coiled titin proteins that are anchoring the myosin to the Z disc or Z line. So what we need to add to this are these troponin and tropomyosin proteins and talk about their relation to getting this sarcomere to actually contract. If we're thinking about contraction, we're going to need to think about actin and myosin actually contacting one another. Typically, actin and myosin are prevented from getting together due to this tropomyosin rope. I'm looking for this picture here. So here's our myosin filament that have those myosin heads like we've drawn on there. Those myosin heads are typically pivoting away from that M line. And if we look up here to what we've drawn, the actin filaments, what I've drawn in kind of an orange color here, they're represented in a blue color on the slide. But if we can zoom into those actin filaments, you can see that active site that I'm talking about, this little depression. Maybe this pocket on my shirt can represent the active site. In this case, I'm actin, and I've got this little active site. Typically, in a relaxed muscle, you can see that this long thread-like protein, that's the tropomyosin rope, it's covering all of those active sites. What's holding this thread-like tropomyosin in place is that rounded protein right there called troponin. Let's see if I can zoom in on that. This is just a zoomed-in picture. Now actin is in a red color, but you can see that thread-like tropomyosin. It's being held in place by this rounded protein called troponin. Troponin is a complex of a few proteins. You can see those two smaller proteins I sometimes like to think of as hands. And again, I think I have just enough room to sketch this weird thing on the board. But what I'm trying to show the difference in is the difference in a contracted muscle versus a relaxed muscle. So let's say here in a relaxed muscle, what we're going to see is, let's take our troponin color. And troponin has those two little rounded hands to it. I'm going to exaggerate those hands to look like this. Troponin is always holding on to two things. And we said its safety blanket is that thread-like tropomyosin rope. So anywhere we see troponin, tropomyosin is going to be drug around. That's even the case. I'm just going to compare this real quick to a contracted muscle. When we're talking about a contracted muscle, once again, it involves troponin and it's two hands grabbing onto stuff. One of those hands is always going to be carrying around this tropomyosin rope. That's the long thread-like rope. If we're thinking about a relaxed muscle, it's exactly what we're looking at here. We can see troponin. It has one hand on that thread-like tropomyosin rope, and the other hand is on actin. And when tropomyosin is held together on top of actin, what happens is tropomyosin blocks those active sites. I'll say it like this. Active sites are blocked by this tropomyosin. So actin and myosin cannot get together. Those active sites, which is the only place myosin can bind to, are covered up by this tropomyosin rope, ensuring that we've got a relaxed muscle. But something happens in a contracted muscle, and it's given away right here for the first time. Calcium ions. If calcium ions show up, it turns out that troponin prefers to hold on to calcium ions rather than actin. I'll let this green color represent my calcium ion. If calcium ions are present, then tropomyosin grabs onto calcium, and in order to grab onto calcium, it lets go of actin. So now, when calcium ion is on the scene, actin is over here. We can say active sites exposed. And the reason active sites are exposed is because troponin has let go of actin. So now you get this actin and myosin will form cross bridges. So this is what leads to a contracted muscle. We've got actin and myosin that are forming these cross bridges because those active sites have been exposed. And again, active sites are only going to be exposed if calcium ions are on the scene. We had mentioned something about calcium ions earlier. They're present in muscle cells. They're locked away in one of those organelles. There it is. There is that organelle that we're talking about, this kind of blue net-looking organelle that completely surrounds these myofibrils. That organelle is called the sarcoplasmic reticulum, and it basically stores large amounts of calcium ions. It actually has these voltage-gated channels that are all along the surface of that sarcoplasmic reticulum. And voltage-gated channels, like their name tells you, if they're hit with an electrical current, they open or close. So these organelles can very rapidly, if they sense an electrical current, they can very rapidly shower calcium ions into this sarcoplasm. That is going to lead to muscle contraction. If we want the muscle to relax, all we have to do is, like a vacuum pump, suck all of those calcium ions out of this sarcoplasm and put them back into that sarcoplasmic reticulum. If we can get rid of the calcium ions, then that's going to lead to the muscle relaxing. I feel like now that we're talking about T-tubules, I'm going to need to get into yet another sketch. But before I do that, I'm just going to hold up this muscle fiber model. We know that this, the collagen fibers, this is telling us the connective tissue that wraps around the outside of a muscle cell. So this would be endomysium. This surface of a muscle cell, instead of calling it the plasma membrane, we call it the sarcolemma. And you'll see that the surface of this sarcolemma has little holes. Those holes we're going to call T-tubules, and if we look through the cross section, they're showing these T-tubules in kind of a red color. We'll learn that T-tubules are the way that electrical currents get deep down into the muscle cells. What those electrical currents do once they get deep down into the muscle cells is they make contact with that sarcoplasmic reticulum and release calcium ions. Here's another picture again of that sarcoplasmic reticulum kind of wrapping around each myofibril. That is the organelle that delivers calcium ions and then removes calcium ions very quickly. There is a term in your lab list called a triad, and you'll notice that I sometimes think of a triad as like a T-tubule sandwich, where I guess the ham and cheese of this sandwich would be the T-tubule itself, and the pieces of bread that make up this sandwich, those are the terminal ends of this sarcoplasmic reticulum. See how the terminal ends of the sarcoplasmic reticulum, they form this continuous connection with the surface of the T-tubule. So that little bit where we have the ends of the sarcoplasmic reticulum making up our slices of bread, and then the peanut butter and jelly or whatever in the middle, that's the T-tubule. Those three things together form a triad. And as an electrical current makes its way down through this T-tubule, it's going to shake open these voltage-gated channels on either side of that T-tubule. So again, calcium ions are going to flood this sarcoplasmic reticulum. There's a lot of words on this slide that I'm about to explain. They describe what happens when we get a muscle that actually contracts. This sliding filament theory, as we see these filaments, the sliding action that actually occurs is these two Z discs or Z lines are going to slide towards one another. I feel like I have that in an animation. Here it is. Notice how the A band itself never changes width. What actually changes when a muscle contracts is the width of those I bands. Let me start it again. You can see when a muscle contracts, those two Z lines are going to be pulled closer together. What's really disappearing here is that H zone. We said an H zone was a place where there was thick filaments only. When a muscle is relaxed, you've got a really large H zone, and that disappears as the muscle contracts. Okay, so what I like about this animation is it shows those Z discs being pulled closer together, but what it lacks altogether is it's not showing you any type of connection right here between actin and myosin. There are these cross bridges that form between actin and myosin, like I just mentioned, that are responsible for pulling these two Z discs closer together. Here's a slightly better picture of that. We can see those myosin heads on the thick myosin filaments. And in a relaxed muscle, we can see a really thick I band. There's a lot of space in that I band. But in a contracted muscle down here, you can see the space in that I band has been greatly reduced. The width of the A band is exactly the same in a contracted muscle versus a relaxed muscle. So those I bands shrink. So back to what's actually causing those I bands to shrink. What's causing this cross bridge to form is really the exposure of those active sites. So right now, this is a situation where we've got what we've drawn right here. We've got a relaxed muscle. We can see that the troponin here is holding that tropomyosin rope in such a way that it's blocking all of those active sites. Here's the myosin head. It wants to form a cross bridge, but it can't do that because the active site is covered up. But then calcium ions show up. Calcium ions are, it's how you say it is, troponin has a greater affinity for calcium ions than it does for actin. So given the choice, if calcium ions show up, it grabs onto one of those and it lets go of actin, temporarily exposing those active sites. We can try this. How many people? Can I get y'all to participate real quick? I don't know if I've got this active site right here, so I'll have to be actin. We need a troponin and a tropomyosin.
You would do that?
Sure. We need some people to participate here. You want to be troponin or tropomyosin? Doesn't really matter. Troponin, right? You're troponin. Then you're going to hold on to tropomyosin. You're going to be tropomyosin for a second. And as troponin, you've got these two arms, so you're going to hold on. You don't want those hands to ever be empty. You're always going to be carrying around this tropomyosin rope. So maybe put a hand on her arm there, and then you can hold my arm. Now I'm going to get kind of close to you just so that you're blocking that active site. So right now, troponin is holding tropomyosin in a way that this active site is covered up. So no cross bridges can form. Justice, do you want to be the calcium ion? Sure. Or anybody? So if calcium ions show up on the scene, what's going to happen is that troponin is going to want to let go of something. Troponin never wants to let go of tropomyosin. Instead, you let go of actin, and you grab onto calcium ions. You prefer to hold on to calcium ions. So as long as calcium ions are there, I've got this exposed active site, and we've got a contracted muscle as long as calcium is there. But if calcium will go back to the sarcoplasmic reticulum just temporarily, you don't want that hand to be empty, so now you're back to holding on to actin, and that active site is covered up. So basically, to expose the active site, we just need free calcium in the sarcoplasm. And if we want to cover that active site back up, we just need to put away calcium ions. All right, thank you.
I have a question. So once you go back, it's relaxed, right?
Say that again. Once this active site is covered up, once calcium ions go back, and this tropomyosin rope goes back to looking like this, where it's covering up those active sites, now we've got a relaxed muscle, because that prevents any actin and myosin forming the cross bridges.
So like a contracted muscle would be like if you get like a cramp or something, or like...
We're almost there. So what happens in that cramp is that you've got these cross bridges that have formed, the power stroke has happened, and you actually need AT. Let me just show a picture of that. We're going there. Okay, so this is just that sliding filament theory. When we're talking about a contracted muscle, those sarcomeres have shortened. They've pulled those Z lines closer together. I feel like the rest of this slide is when we get into the next and final diagram. This is actually where we split between muscle tissue and our neuromuscular junction, because so far we just talked about this part. We've drawn skeletal muscle fibers. We know the thick and thin myofilaments that make up this myofibril, this contractile organelle. We know in order to get this organelle to contract, we just need to get calcium ions to be released into this sarcoplasm. Calcium ions are going to come from that sarcoplasmic reticulum, and they become released if hit with an electrical current. So here's where we find out where that electrical current comes from. It comes from a motor neuron. We've erased it, but we said these mechanical events of contraction and relaxation are always preceded by some type of electrical event. So here is the electrical event that is going to precede contraction of this muscle. And to kind of give it away real quick, we're going to, in fact, I think it might just be right here. This might be more helpful than me just using my words. This is the graph that shows electrical events precede these mechanical events. So a mechanical event like contraction or relaxation of a muscle, is always preceded by some type of electrical event. This electrical event, you can see, once it happens, there are a few milliseconds where there is no contraction at all. This time period where the electrical event has occurred, but there has not yet been any mechanical events, is what we call this latent period. To go back to the previous picture, you can see what is causing that latent period is this electrical signal hits the muscle fiber, but after that electrical signal has occurred, it takes a few milliseconds for that electrical signal to move down the surface of the sarcolemma, down into this T-tubule, and open up those voltage-gated channels on the sarcoplasmic reticulum. Then a few milliseconds for that calcium ion to diffuse into the sarcoplasm, and then get the active site exposed before you can finally get contraction to occur. To go back to this slide, this increasing strength of contraction corresponds with increasing numbers of calcium ions in the sarcoplasm. With the maximum number of calcium ions, we get the maximum number of exposed active sites and the stronger contraction. As this relaxation is occurring, what's basically occurring is the reduction in free calcium ions in the sarcoplasm. When all of those calcium ions have been put back into the sarcoplasmic reticulum, then all of those active sites are covered back up and we've got a completely relaxed muscle. So again, latent period has to do with the fact that it takes a few milliseconds after that electrical event occurs before there are ever any calcium ions that get released. And as those calcium ions accumulate in the sarcoplasm, we get more and more of those cross bridges, a stronger contraction. And in order to get the muscle to relax, like I said, we have to put away all of those calcium ions. Okay, I feel like we're going to end up drawing this at some point. Let me get through a few more slides and we'll see if I get to draw that today or not. But stop me at any point if I'm stirring up questions at all. This slide introduces us to the term motor unit, which we can think of as a motor neuron, one of these motor neurons here, and all of the muscle fibers that it goes to. For example, we've got two motor units here. One motor unit is shown in kind of a blue or purple color. They're calling this motor unit number 1. You can see how this is a motor neuron that goes to just 2 of these muscle fibers. So that's motor unit 1. We can see, just as an example here, we have another motor unit, what they're calling motor unit number 2. They're drawing it in a red color. Notice how motor unit number 2 is going to completely different muscle fibers. These different motor units are one of the things that are allowing you to make such fine movements with your hands and fingers. So they're controlling different muscle groups, different muscle fibers. And just checking that this isn't on there, but if you are lifting things that, like if I'm just lifting this marker, I don't need to use all of my muscle fibers to lift this. It's not very heavy. So maybe motor unit number 1 with just those 2 muscle fibers is strong enough of a contraction for me to lift this marker up. But if I wanted to pick up something like this model that's heavier, I would need to recruit more of these muscle fibers. I couldn't do it with just 2 muscle fibers. I might need 5 muscle fibers. So motor unit number 1 and motor unit number 2 can fire at the same time and give you a greater strength of contraction. You're just firing more of those muscle fibers at the same time. Okay. Again, we're just looking at motor neurons that send these electrical signals to muscle cells. And we know muscle cells respond to the electrical currents. There's that electrical stimulus that always precedes these mechanical events. This is the graph of the muscle twitch. A muscle twitch, we can see, is just a single stimulus followed by this contraction, relaxation. If there's just one stimulus, that short little shock is going to cause a burst of calcium ions to be released. Again, when the calcium ions are at their maximum, number, that's when we have the strongest contraction. And if there isn't any more electrical events that are coming behind there, just one electrical event, then no more of those voltage-gated channels are going to open. Calcium ions instead are going to be put back into that sarcoplasmic reticulum. As we lose free calcium, we lose that strength of contraction. I didn't skip over anything there. We talked about motor units. We talked about latent period, contraction and relaxation.
Can I ask a question?
Sure, yeah.
Sorry. On
Throw
here
it.
number 3 says thick myofilaments are made of the protein. Do we put myosin?
Thick myofilaments are made of the protein myosin. That's right. Thin myofilaments are made of actin, which bonds to the thick myosin, which bonds with the thick myofilament proteins. Then you have blank, which bonds to calcium ions. So the next blank is troponin, which bonds to calcium ions and tropomyosin. Number 4, that calcium is stored in the sarcoplasmic reticulum. Number 5 is T-tubules. Yeah, let me know if I skip over some of those.
Can you go over 3 again? Can you repeat that?
Yep, number 3, thick myofilaments are made of the protein myosin. Thin myofilaments are made of the proteins actin, which bonds with the thick myofilament proteins. So actin and myosin are the only two that bond together, the thick and the thin myofilaments that bond together. After where it says bonds with thick myofilament proteins, the next one is going to be troponin, which bonds to calcium ions, and it also bonds to tropomyosin.
So number 2 would be don't tell me what it is. That's right.
It's this thing here, the sarcomere. So those sarcomeres actually shorten during contraction. That's that functional unit. It's the link in that chain.
Okay.
So the chain is the contractile organelle, and the functional unit of that chain is the sarcomere.
Okay. Thank you.
Yeah, no problem. No problem. Number 1, one skeletal muscle cell is called a muscle fiber. Remember, muscle cell and muscle fiber are the same thing. So the cytoplasm, instead of calling it cytoplasm, we call it sarcoplasm. And instead of referring to one of those organelles as the endoplasmic reticulum in the muscle cell, we call it the sarcoplasmic reticulum. Wherever that was. There's the sarcoplasmic reticulum.
So on number 6, where the second part, it says in the myofibril, the band only connects thin myofilaments. Would that be Z band?
So in a myofibril,
Or I mean, not Z, A band?
the I band only contains thin filaments. So I band is thin filaments. So number 6, it says one sarcomere extends between 2 Z discs. In a myofibril, actin overlaps the myosin in an A band, one of those dark bands. Then below that, in a myofibril, the I bands only contain thin filaments. So we have titin and actin there. The next below that, it says it should say myosin. The myosin filaments attach directly to the M line. So we got myosin attached directly to the M line, and then we have the thin myofilaments attached directly to the Z line. And then the last one there, titin molecules, are those spring-like proteins. Do we feel better about that one?
And 4 was sarco, what again?
Calcium is stored in this sarcoplasmic reticulum.
Okay.
Here it is. Sarcoplasmic reticulum is this blue structure. Does everybody feel good about that first page now? A little better?
Does this only happen in smooth ER? Like it says network of smooth ER. So not in each mile. Like this only happens in smooth ER.
Yeah, so going back to like a biology class, like when we talked about this endoplasmic reticulum, there are some parts that are smooth and some parts that are rough. The rough part indicates that there are ribosomes present, and ribosomes indicate that you're building proteins and you're packaging stuff. Smooth endoplasmic reticulum, there's no proteins, so you're not building anything. And that's right, we're not building anything here. We're just storing calcium ions and releasing them. No problem. What are you thinking?
Muscle fiber. And the third one, endoplasmic reticulum, is called G.
Endoplasmic reticulum is called the sarcoplasmic reticulum, right here. That's the third line on number 1. So part of it is just naming the parts of the cell with the correct weird muscle word.
Well, yeah.
Did I answer your question, or am I still?
No, I was just gonna ask about page 2. I don't know if people have.
Yeah, so page 2 looks something like this, where we've got an image that looks something like that. One of those letters is pointing to the T-tubule. Another letter is pointing to the surface of the cell, this sarcolemma. There's another letter that's pointing directly at this webbing, this sarcoplasmic reticulum. And then you got letters F and G down there. One of them is trying to indicate a I band, and the other one is indicating a band, a dark band. We feel better about the second page now? As I'm flipping through here, we should be able to do page 3 and even page 4.
So what was which on the diagram, the T-tubule?
So our T-tubule, well, just to go to this one, that structure is this T-tubule.
Okay.
It runs perpendicular through the muscle fiber. It takes electrical current deep down into the muscle fiber where that electrical current makes contact with the sarcoplasmic reticulum.
So according to the diagram within the packet, so the A band, which is your thick band, which is G, your thin band is F, and your T-tubule is C?
That's correct.
Okay.
So letter B would be the mitochondria there. And can you see where letter A is just trying to point to one of these myofibrils? It's trying to show that that's the contractile organelle. This myofibril is surrounded, it's cloaked by this sarcoplasmic reticulum. And each myofibril, remember, is made up of those thick and thin filaments, which we can kind of see there as the thick dots and the thin little dots. And that's what we drew over here, those thick and thin filaments. All right. Still feeling all right? I'm looking at page 4. I'm just going to catch up to this picture right here. And on page 4, this is what we should use this diagram to answer page 4. One of those letters is pointing to this thread-like tropomyosin rope. Another one of those letters is pointing to this rounded troponin protein. And then another one is pointing to the kind of double layer of proteins there that represents this chain of actin proteins. And then you've got parts of the myosin down here. There's the thick myosin filament with the myosin heads, always pivoting away from that M line in a relaxed muscle.
How do we know, like, what the difference between, like, on the diagram, like ours, because ours is just black and white, how do we know, like, the difference between troponin and actin? Like, is one always going to be a different color?
That's a good question. So tropomyosin is always going to be this thread-like protein, and actin is going to be a double layer of these.
Well, like troponin and actin, like for us, we just have like a black and white circle.
Yep, so you see how there's like a continuous chain of white circles? Mm-hmm. Those represent the actin. And occasionally you see those troponin proteins that they're not a continuous chain of troponin, they're just kind of scattered every so often, and they're keeping that tropomyosin rope covering up those active sites. Yeah, no problem. So in yours, kind of that would be letter C that's pointing to the troponin.
Mm-hmm. And then the actin would be B?
Correct.
Thank you.
Yeah, no problem. What I'm trying to get to is what you were talking about, which is when the active sites are exposed, like they're doing right now, the first thing that happens is a cross bridge will form between actin and myosin. And when that cross bridge forms, you can see how the myosin head is still cocked back. Each of these myosin heads, we have kind of pivoted away from the M line, and what that is analogous to is, if you can think of like a revolver or something like that, one of these handguns that you have to cock a hammer back in order for the thing to fire. These myosin heads are just like that. And in order for them to fire, they have to be set. They have to be cocked back in this loaded position. And when these myosin heads are pivoted back like this, they're locked and loaded. They're ready to fire. All they have to do is attach to that myosin head, and then you can see the next thing that's about to happen. This myosin head is going to pivot. Let's see if we're to that part yet. Let me get just a little bit further through this, and then we can take a little 5-minute break if we need to. The next few slides, in fact, actually talk not necessarily about the anatomy of this, but just about the physiology of muscle tone. We'll use this term muscle tone more than just today, but what you can think of when you're thinking about muscle tone is this slight, constant contraction that muscles do. A lot of it is often just to help maintain body posture and position. And I mentioned, like, when people have those collisions, like on the football field, where somebody gets knocked out immediately, they lose that muscle tone, and that's why they look like a limp noodle that hits the ground. So again, slight constant contraction is what we call this muscle tone. Later on, we'll talk about sympathetic and parasympathetic muscle tone. You've got, for example, your heart would want to beat about 100 beats per minute on its own if it weren't for the fact that you've got this slight, constant parasympathetic signal going to the heart telling it to slow down. So it beats at, if on average, a normal heart would beat about 76 beats per minute with that slow, constant parasympathetic signal that is telling the heart to slow down. If we think about muscles, whether it's the heart muscle or whether it's a skeletal muscle that you're using to exercise, muscles are going to have a very high demand for energy. And there's a few different ways in which they can get their energy. We'll go through these 3 different ways in which ATP is derived by these muscle cells to get them around. The first way, we'll see all of these in order. The first way in which ATP is made by the cell is by direct phosphorylation of ADP. We mentioned ADP and ATP earlier in the semester. This is an adenine nucleotide that has 2 phosphates attached to it. If we stick a 3rd phosphate on there by ripping a phosphate away from a creatine phosphate and sticking that phosphate onto an ADP, we've created an ATP. It's a very quick process. It is a single step. It just doesn't produce a lot of ATP. We'll see there's another way in which we can produce ATP, which is called glycolysis. We simply take a glucose molecule and karate chop it in half. So it is a 1-step process that yields some ATP. The 3rd step down here, aerobic respiration, this 3rd step is the only one of the steps that actually requires oxygen. Maybe the other note that you can put down here, in addition to requiring oxygen, it's going to require quite a bit of time, like on the order of minutes, whereas these other 2 processes that produce ATP do it like on the order of seconds. But it's just not a lot of ATP. So you've got rapid bursts of small amounts of ATP, but given enough time, aerobic respiration can give you large amounts of ATP. Here's an analogy to see what I'm talking about. Think of somebody that's doing like a 40-yard dash. If you're a good sprinter or something like that, in a 40-yard dash, this might take you 4 or 5 seconds to run across those 40 yards. Those 4 or 5 seconds, that's not enough time for you to take in a deep breath and let that oxygen get to all of the cells and circulate around. When you're doing these sprints, it's the first thing that you use, your muscles are going to deplete any ATP that's currently available. So they used what's on hand. That's depleted within the first few seconds, and then your body has to produce more ATP just instantaneously by doing things like ripping a phosphate off of a creatine phosphate, sticking that extra phosphate onto an ADP, and kind of quickly creating an ATP molecule. That instant production of ATP from creatine phosphate, again, only lasts for a few more seconds. If you're still running past 10 seconds in one of these sprinting type situations, your body then has to start to break down glucose anaerobically without the help of any oxygen. You don't need oxygen to karate chop a glucose molecule, so that's helpful. It does yield a couple of ATP molecules every time you karate chop a glucose. So this is yet another way that you can produce small amounts of ATP. But if you're doing like a marathon, somebody that's going to be running for hours, those marathon runners, they're not immediately sprinting out of the gate. It's more about establishing a certain pace. And they're allowing time for their body to breathe and use that oxygen to generate large amounts of ATP. So we saw a couple of pictures earlier that individuals that are good marathon runners typically have a high amount of these slow twitch muscle fibers. Slow twitch muscle fibers, again, these are dark staining fibers due to the concentration of mitochondria. Some of these muscle fibers lack that color because they lack mitochondria. I'm going to go back to the slides I just skipped over. Remember, these cells that don't have any mitochondria, they can't ever do this last step of aerobic respiration and produce large amounts of ATP. Without that mitochondria, the only way those muscle cells can produce ATP is either by breaking down glucose directly, this anaerobic pathway, or we said directly by making ATP by ripping a phosphate off of a creatine phosphate and attaching it to an ADP, basically taking this dead battery and charging it up to make it a charged up battery. So direct phosphorylation of ADP is one way to create new ATP. Karate chopping glucose molecules is another quick way of making ATP. Neither one of these require oxygen or much time at all. But to get to the large amounts of ATP, we're going to need mitochondria, we're going to need oxygen present. So that, again, takes a little bit of time. And fast twitch muscle fibers, again, without that mitochondria, they're only going to be able to produce short bursts of ATP. People that are good sprinters, 40-yard dash types of athletes have a higher concentration of these fast twitch muscle fibers, which again allows them a greater access to ATP out of the gates, but aren't going to be able to produce long or prolonged amounts of ATP due to the lack of available mitochondria. So here are the labeled slides. Slow oxidative fibers, those are the ones dense with mitochondria. Fast glycolytic fibers, these are the ones that can only go through glycolysis. They can karate chop a glucose molecule, but they can't do any aerobic respiration. So fast glycolytic are the light ones. Slow oxidative, again, these are the ones that are using oxygen. They have a bunch of mitochondria, so that waiting for mitochondria to use oxygen to break down glucose takes a long time, but you get a lot of ATP out of that. The intermediate ones are these fast oxidative fibers, where they can still produce fast bursts of ATP, but they're also oxidative. They've got a little bit of mitochondria. We can see that with the dark color. Those mitochondria are going to use oxygen and, again, produce large amounts of ATP. I skipped over this slide, which is just what I was saying there with words. The speed of contraction is what we describe as either fast or slow. And then those metabolic pathways are where they get the term oxidative. If they're using oxygen, they have to have mitochondria present. If they lacked mitochondria, that's what we saw, those glycolytic fibers. Glycolytic, no mitochondria whatsoever, but if they do have mitochondria, then they're oxidative. Let's see. So we introduced some muscle fibers there. We talked primarily about how a muscle fiber contracts, really these sarcomeres. And here is where I I left off. I said I felt like another diagram was coming soon. And it's actually the diagram from our neuromuscular junction PowerPoint. It's only like, I don't know, maybe 5 or 10 slides, and it's all about this neuromuscular junction. But since I've been talking for a while, if we need to take like a 5-minute break or so, let's do that. All right. I haven't done this one in a while, so here goes. Stop me at any point if there's questions lingering from anything else. But what we're going to attempt to do here is sketch a neuromuscular junction. So this is just where a neuron and a muscle meet. We know that the neuron tells the muscle what to do. And you have a diagram. This is the very last diagram on the back of your lecture handout. It shows you something like this, what I'm holding up. This is a skeletal muscle fiber, or what we call a muscle cell. And we know this is endomysium around the outside of the muscle fiber. This is the plasma membrane of the muscle fiber, what we would call the sarcolemma. And if you look around this sarcolemma, right there, you can see there's a rough patch on this sarcolemma. And that is also where this motor neuron is coming down. And it's not just touching anywhere on this muscle fiber. That motor neuron is touching right there on that little rough patch. On this model, those are touching together. Just as if you give me like a high five right here. Notice our hands aren't really touching, but they're really close together. That little space in between our hands is the synapse. And the neuron and the muscle are going to be just like our hands. They don't actually touch, but they get really close together. So I'm going to put that up here on the board. In fact, I'll start with my muscle fiber. I'm leaving this up because I want us to remember this is going to be inside the muscle cell. Let's just try it this way. Let's take this thing, and I'm going to have to leave myself a little bit more space. I'll start it right there. Let's start right here and go a little ways. Then I'm going to make this little shallow depression. And we're going to carry this on down. This thing that I'm trying to draw here, this is our muscle cell. Muscle cell or muscle fiber. Labeling parts of the muscle fiber. We know that this smooth plasma membrane is what we call the sarcolemma. That's just the plasma membrane of this muscle cell. And if we follow this muscle cell all the way down, let me take it a little bit further and cross over into this area. I'm going to label that structure as one of our T-tubules. There's our T-tubule. It's going to be this depression in the sarcolemma that takes us down into the center of this muscle fiber. And what it's going to contact down there is this sarcoplasmic reticulum. And what we know about that sarcoplasmic reticulum is that it's full of calcium ions. Let's put those calcium ions in this color. So sarcoplasmic reticulum full of all of those calcium ions. There are little voltage gates on the edge of that sarcoplasmic reticulum and the T-tubule. And there's a sarcoplasmic reticulum on either side of this T-tubule. Full of a bunch of those calcium ions. And what I haven't drawn really inside the muscle cell, but what we know is going through the muscle cell are those contractile organelles. These myofibrils are all throughout here. What we've drawn so far, we've got our T-tubule, we've got our sarcoplasmic reticulum, we've got this rough patch that is part of the sarcolemma that we're going to call the motor end plate. Motor end plate is the part of the muscle fiber that there's this shallow depression. But what's also unique about this motor end plate, it's a shallow depression that contains receptors for neurotransmitters. So these little receptors down here, let me say something about those receptors. I'm just going to take that little receptor that I'm trying to draw right there, and I'm going to label this as a chemoreceptor. This is a receptor. It docks chemicals. Let me say that a different way. I'm going to call this a chemically gated channel. Chemically gated channels, they open and close based on the presence of a chemical signal. These chemically gated channels, like I said, are only located on the motor end plate. I'm going to compare these chemically gated channels to something I'm going to draw up here on the sarcolemma. Once we get out of this motor end plate and up here on the surface of the sarcolemma, all along the surface of this sarcolemma, we're going to see voltage-gated channels. And these voltage-gated channels are going to be specific to certain ions. The green one that I'm drawing here, we're going to let these represent sodium voltage gates. And I'm going to use a purple color here to represent potassium voltage gates. Basically, we've got gates that open and close based on a voltage, or gates that open and close based on the presence of a chemical signal that are there. The sarcolemma all the way down to the T-tubules are going to be where we find the voltage gates. All the way down into that T-tubule, we've got voltage gates for potassium and sodium. Again, they open and close based on charge. Only here in the motor end plate do we have these chemically gated channels. Okay, so we've kind of set everything up, and we know in this, what we've basically built here is a stack of dominoes. And if we can get one domino to fall, these voltage gated channels, kind of like dominoes, if we can get one of them to fall, the rest of them are going to fall, and it's going to end up triggering a signal to this sarcoplasmic reticulum. If we can get a charge down here to the sarcoplasmic reticulum, we know it's going to lead to the calcium ions flooding out of that sarcoplasmic reticulum into this fluid of the muscle cell that we're calling the sarcoplasm. If we have a bunch of calcium ions in the sarcoplasm, that's going to lead to the muscle contracting. If we don't have any free calcium ions in the sarcoplasm, all of those calcium ions are stored in that sarcoplasmic reticulum, well, now we're going to have a relaxed muscle. So it's all about releasing those calcium ions, and now that we've kind of set up the part of the muscle, let's add the neuron here. And I don't have a different color green, so let's just put this thing in green. This is going to be our axon. It's just part of the neuron that is sending a signal away from the neuron's cell body down towards this expanded end of the axon, what we call the axon terminal. That axon terminal is actually going to be filled with vessels. This is a vesicle. Once again, we know cells package proteins up in these little vesicles. These vesicles at the ends of neurons are going to contain neurotransmitters, these chemical signals that are produced by neurons. We'll talk about a bunch of different types of neurotransmitters by the end of this. This is a vessel. Now it's got neurotransmitters.
So the axon, that's the name of the whole thing, or just like the little pathway?
The axon is just the part of the neuron that's sending signals away. In fact, if I give you a full sketch of a neuron, I typically draw a neuron like this. Where there's our axon terminal. Kind of that expanded end of the axon. This would be what we would consider the cell body. Let's just label it this way. I know we're not quite to neurons yet, but if we're thinking of a typical neuron, that bit is the cell body. These little root-like structures that stick off we're going to call dendrites. And then this thing right here that's sending electrical currents away from the cell body all the way down to the axon terminal, this part of it is what we call the axon. Okay. So now all I've drawn is just the axon and axon terminal of a motor neuron that's coming down to talk to this muscle fiber. We want to know what a motor end plate is. It's part of the muscle fiber that contains those chemically gated channels. And what docks to those chemically gated channels are the neurotransmitters from this neuron. So right now we've got a relaxed muscle, nothing has happened. But if we're going to get this muscle to contract, we're going to have to basically get calcium to release from that sarcoplasmic reticulum. We've set the stage. Have I stirred up any questions so far with our diagram?
Well, for us on number 12 for ours, our receptor would be the little triangles.
Oh yeah, let's look at that diagram. So on your diagram, you've got something that kind of looks like that. The receptors are those little triangles. That would be letter E.
And then the actual end plate would be D.
Yeah, that didn't really come out very well on the copies that I made, but that's correct. Letter D is trying to point to that motor end plate. And then A would be that axon terminal. B would be the synaptic vessel with acetylcholine. And the only thing that I forgot to label here, even though I tried to show that with justice here, was that space in between where the neuron and the muscle, they don't actually touch, but that little space in between is what we call our synaptic cleft. And that's what we're about to talk about. This space, since the neuron and the muscle don't actually touch, what really makes this journey across that space are those neurotransmitters. But since I said this was, that's actually as detailed as it gets, all we're going to do now is just list the sequence of events that occur. And I'm going to have to erase this if that's okay. You thinking about something? All right. Looked like you were. All right, I'm going to list in order basically what we're going to see occur between a neuron and a muscle. Then we're going to catch up with those slides. I guess that's as good as I can do with that. Okay, so here's how this works. We're going to see this in an animation and in a video in just a second. But the first thing that occurs in order to get this neuron to talk to a muscle, let's just put number 1 up here. We're going to say an action potential, which I'm going to abbreviate AP. This action potential, it's another way of saying an electrical charge that moves down the surface of this axon. So this is electric current. In fact, jumping ahead to Unit 4, we graph an action potential that occurs on the surface of a neuron. We talk about this wave of electrical activity that goes from the axon all the way down to the axon terminal. So stepping aside from the details of an action potential just for a second, we know it's an electrical current that is going to make its way to the axon terminal. So let's say that action potential arrives at axon terminal. What that action potential is going to do when it hits that axon terminal, here's where I don't have enough colors, but I'm going to use an orange color to show that right here on the axon terminal, we're going to have, I'm just going to put them in an orange color, and I'll label what these things are because I haven't given myself a lot of space. Those orange structures are going to be voltage-gated calcium channels. So voltage-gated calcium ion channels, those are located on the axon terminal, and they open and close based on the presence of a voltage. So Once that action potential, which we know is a voltage, arrives at the axon terminal, the second thing that it does is it opens those voltage-gated calcium channels. So opens voltage-gated calcium ion channels. What we will see, I'll just give this away right now, there are currently more calcium ions outside of this cell than there are inside that neuron. So when these voltage-gated calcium channels open up, calcium is going to move into the cell. So opens voltage-gated calcium ion channels, and I'm just going to squeeze at the end of this, calcium ions move into cell. They move into that axon terminal. Now there's something that corresponds. As calcium ions move into this axon terminal, they actually start to surround these vessels. Now again, for details, for reasons that I have not yet detailed, these calcium ions, they each carry a positive charge around them. What we will learn, and I know I'm drawing this so small that it's hard to see, neurons. When we look at the plasma membrane of a neuron, if we're inside this neuron and we get really close to the plasma membrane, we're going to see that the edge of that plasma membrane of a neuron from the inside is negatively charged. It's got this wall of negative charge around the inside of that neuron. These plasma membranes, these vessels, they have negative charges around them. And these negative charges repel one another. So these vessels just kind of float around in the middle of that axon terminal until calcium ions move in. These positive charges are attracted to the negative charges. So calcium comes in and it surrounds these vessels with positive charges, which are now attracted to the negatively charged plasma membrane of the neuron. What I'm trying to say here basically, as calcium ions come in, that's what is going to cause these vessels to move all the way out to the surface of this neuron. They're going to merge with the plasma membrane, and they're actually going to drop all of their contents into this synaptic cleft. So this neurotransmitter, let's just say this neurotransmitter is acetylcholine, because that's the one that they use as an example in the book. So this would represent our acetylcholine, or neurotransmitter that's released, and it takes a few milliseconds for this neurotransmitter to diffuse down to the receptors on the motor end plate. So far what we've seen, action potential arrives at the axon terminal. It opens those voltage-gated calcium channels. As calcium moves into the cell, right after calcium moves into the cell, we can see vessels are going to merge with the plasma membrane. We'll say vesicles with neurotransmitter, this acetylcholine, merge with plasma membrane. After they merge with the plasma membrane, I'll just put number 4 down here, they release that acetylcholine into the synaptic cleft. So as that acetylcholine is released into the synaptic cleft, I'll just put to the side of this over here, it basically diffuses across to the receptors on the motor end plate. So it takes a few milliseconds for that acetylcholine to diffuse across to the motor end plate. But here's the thing, on number 5, as acetylcholine docks to those receptors, these receptors on the motor end plate, these are, I've just drawn them as a little stick right here. But in reality, these receptors, let's see if I can draw it another way real quick. I'll say as acetylcholine docks to those receptors, it causes these chemically gated channels to open up. As this acetylcholine docks to receptors, on that motor end plate. Once the acetylcholine docks to receptors on the motor end plate, I'll just keep it right here on number 5. It's going to cause these chemically gated channels to open. I guess I can just put this over here. These chemically gated channels, I'm just going to circle this and give this another descriptive term. These are going to be what we call non-specific ion channels. Better. And we're going to talk a lot about chemically gated channels versus voltage gated channels. And it's typically the case that these chemically gated channels are non-specific ion channels. They don't care what ion crosses through. They're not specific. Versus these voltage gated channels that we have seen are very specific. They either let certain ions like sodium cross and nothing else, or they'll let potassium cross and nothing else, or they'll let calcium cross and nothing else. Chemically gated channels doesn't care. They're going to open and close based on the presence of a chemical signal, and ions are going to be able to cross in either direction. So back to over here. Once acetylcholine docks to those receptors on the motor end plate, this chemically gated channel is going to open up. And since it's a non-specific ion channel, I'm trying to find my colors here, our purple and green color, this is what happens right here in number 5. These chemically gated channels open up. We're going to see that sodium is going to move into the cell and potassium is going to move out. So through these non-specific ion channels, anytime they open up, we're going to see sodium moves into the cell, potassium moves out, but there is way more sodium moving into a cell than potassium is going to move out. Both of those are positively charged ions, so you would think that if we had the same amount of sodium coming in as potassium moving out, the charge of the cell won't change. That would be the case, but we have way more sodium ions moving into the cell than potassium moving out. So what that causes right here in this motor end plate, I'll just try and draw this and catch up with my words. What we're saying is this acetylcholine comes down here, it docks to these receptors on the motor end plate. When that happens, it causes this little space, this little channel to open up. Put a little neurotransmitter on that one. When these channels have opened up, as long as that chemical signal is there, these gates are going to be opened. That allows sodium ions to move into the cell, carrying their positive charge with it. And it is true that there may be a potassium ion that leaves, but we're going to have more sodium come in than potassium come out. So what I'm trying to depict right here in this motor end plate, what we have, this thing, this is a wave of depolarization. Right? Depolarization, we said that is a term that describes electrical events. It's going to describe when a cell starts to become more positively charged. It's describing that the cell is taking on positive charges. In this case, this wave of depolarization is due to sodium ions coming in through this chemically gated channel. Now here's the thing with chemically gated channels. If there is sufficient amounts of acetylcholine here that sits here long enough that allows enough sodium ions to rush in, this wave of depolarization, this positive charge, if it happens to get out of this motor end plate and touch our first voltage gated channel, then this is the series of dominoes. We'll make a distinction between these chemically gated channels and these voltage gated channels. These are going to be something that we have to draw a little bit more closely. Sometimes these voltage gated channels are described as like an all or nothing reaction. So basically, if we can trigger one of these voltage-gated channels, that signal is going to make its way all the way down into the T-tubule to that sarcoplasmic reticulum. This thing that I'm trying to draw right here, this squiggly little line, this is sometimes what's called end plate potential. Right? EPP. End plate potential is basically determining whether or not this signal down here in the motor end plate is sufficient enough to get out to the sarcolemma to actually cause the muscle to contract. Has anybody ever seen somebody that does like the reflex test? If I'm sitting here and I'm just lightly tapping on my patellar ligament right there, this very light tap is not sufficient to cause a muscle to contract. Right? So I can feel that I'm tapping on my patella, but this little light tap is not sufficient, so that is causing some sodium ions to move into these motor end plates. But if it isn't a sufficient enough stimulus that enough sodium ions come in to trigger these voltage gates, then it'll never get down to release calcium ions and cause the muscle to contract. If I can do that a little bit harder, I can involuntarily, there goes a good one, I can involuntarily get this thing to contract. And all I'm doing is hitting it with a sufficient stimulus somewhere in there. I've got to not think about it. But anyway, you can see a very light stimulus is not sufficient, but something that is strong enough might get a contraction to occur.
So all of this happens just to contract your muscle?
That's right. If you're thinking like if you want to wiggle your big toe or something, then there's a neuron in your brain that sends a signal down to the muscle that controls those fibers in the big toe.
So the neuro-muscular junction is your neurons telling your muscle what to do?
That's right. That's exactly right. You know how I was talking about earlier, like if you're in, we used to do this in high school where we would grab those electric stim machines and just put them on people for fun. It's not something you should do, but it just shows that muscles will respond to an electrical current. It'll get them to contract. So we're thinking, how do these muscles get their electrical currents? And they're all delivered by some type of motor neuron. So we see electrical events precede these mechanical events. That muscle's not going to contract on its own, but something has to happen to release these calcium ions. Now, you said something earlier, and I'm going to get to that point earlier. What we didn't talk about at all in here so far is we know that in order to get a muscle to contract, we need to get these calcium ions out of the sarcoplasmic reticulum. Now, I'm not going to try and draw this. I'm just going to try and say this with words. So would everybody agree? Right now, we've got calcium ions locked inside this sarcoplasmic reticulum. We've got them completely packed full in this sarcoplasmic reticulum. We can't fit any more of these calcium ions in here. So what we have, what we have created here is what we would call a concentration gradient, where there is a bunch of calcium ions on one side of this membrane and very few calcium ions on the other. So you can imagine that if I were to open up a bunch of voltage gates all along this sarcoplasmic reticulum, initially, which way would those calcium ions want to move? They would want to move out. Just like water in this, if I dump that over, there's not a lot of water in that, but water would want to spread out to an area of lower concentration. So you're right, these calcium ions would at first start to diffuse out into this sarcoplasm very readily at first. But then what starts to happen is about halfway when we get to the situation where there's about as many calcium ions outside of the sarcoplasmic reticulum as there are inside that sarcoplasmic reticulum, we've lost that concentration gradient. So calcium ions are not going to want to flow as quickly. That's not as bad. We've got enough calcium ions here that we can cause contraction. But really what happens is when we want to relax this muscle, in order to get the muscle to relax, we've got to put away all of these calcium ions. We've got to get them to go back into this sarcoplasmic reticulum. And at first, if there's a bunch of calcium ions out here and not very many inside, they would want to move into that sarcoplasmic reticulum. But the same thing happens when you're putting calcium ions back into the sarcoplasmic reticulum. You reach this spot where there are as many calcium ions in the sarcoplasmic reticulum as there are outside. And for the rest of these calcium ions, in order to put them up, you're kind of going against the concentration gradient. And this is we didn't put this in here, but there are, surrounding this sarcoplasmic reticulum, there are going to be what we call calcium pumps. And these calcium pumps, they use a lot of ATP to push these calcium ions against their concentration gradient. They're taking calcium ions from an area where there are very few calcium ions and forcing them against their concentration gradient, packing them back into this sarcoplasmic reticulum. Anytime we're going against the concentration gradient like that, it's going to use a lot of ATP. There's that word again, ATP. So we need that whenever the cell is doing work. Here's a better analogy. Like I lived in San Marcos for a while when I was going to school, and I loved getting in an inner tube at the end of the day, jumping in like right there in that Sewell Park, and then just letting the water carry me downstream to an area of, when you're going with the current, it doesn't require you to paddle or do anything, expel any energy. You're going with the flow of things. And that's how these calcium ions were originally when they were coming back out. And even when they were going back in, originally there weren't very many calcium ions in there, and they were able to flow easily back into the sarcoplasmic reticulum. But the last few calcium ions, they're going against the current. It would be like me putting my inner tube in, like down at the end of the river and paddling upstream. I could do that, but I would need to expend a lot of ATP, a lot of the energy. Somebody said, I think you were asking the question about fourth quarter of a game or something like that, and somebody comes up with cramps or something. You've got a muscle that's in a constant contraction and you can't get it to relax. What you're running into a problem there is you're running out of ATP. You're running out of available immediate ATP to run these calcium pumps. So without this ATP to put away that excess calcium, you have some calcium ions floating around in this sarcoplasmic reticulum, and that's what causes that muscle to lock up and stay contracted.
Okay, so my second question, sorry. So you said electrical events precede mechanical events. So the electrical events are all that down there.
Electrical events are, yes, that's depolarization and repolarization, things that happen.
And then the mechanical event would be the actual action of it happening.
Contraction and relaxation.
But the polarization and the depolarization, that's like all that has to happen before anything can contract, is what you mean.
That's right. Depolarization and repolarization, those are talking about ions moving into or out of the cell, causing that cell to become more positively charged, or if it were losing those positive ions, the cell would become more negatively charged. Right now it's gaining these positive charges, so we're saying it's becoming more positive. We associate this with depolarization. If it was purging itself of those positive charges, we would call that repolarization. So we're kind of getting exposed to these electrical terms too. This is where it seems, maybe the first time we've talked about these things, but we use this vocabulary going forward, so it will become more normal. Let me catch up with what I've written over here. Right now we've got acetylcholine that docks to this motor end plate. We've got sodium moving into the cells, some potassium moving out. More sodium moves in than potassium moves out. So we have this wave of depolarization that, if sufficient enough, if it's a strong enough wave, it might get out of this motor end plate and trigger these voltage-gated channels. Here, I'm going to throw another word out there. End plate potential. We're going to see this as a type of graded potential. Graded potential just means that we are involving chemically gated channels, and the strength of this signal can vary. Let's use this analogy. Imagine that we're standing on a dock. It's nighttime, or I guess it doesn't have to be night. We're standing on a dock near the surface of this water, and there's no wind at all, so it's just like glass on the surface of the water. If I had a sack of a bunch of different size rocks, you can imagine if I took a small little pebble and was throwing that into the surface of the water, it would cause these slight little ripples on the surface of the water. That wouldn't be as strong of a disturbance as if I took like a big boulder and threw that into the water. That would cause a big disturbance in the water. Those varying disturbances, Graded potentials describe those types of varying disturbances. So if we were throwing those tiny little pebbles, that slight disturbance isn't going to cause a big enough ripple to ever get out of this motor end plate. But if we were throwing, big boulders, then that might be a sufficient enough disturbance that electrical charge could get out of the motor end plate. These are larger waves. They might actually disturb some of these voltage-gated channels. Anytime we involve these voltage-gated channels, we're going to be talking about an action potential. Action potentials involve voltage-gated channels. Graded potentials involve chemically gated channels. That type of stuff is all that we talk about in unit number 4. Graded potentials versus action potentials. But we also get exposed to chemically gated channels for the first time and voltage-gated channels. They're not located in the same place. Going back to this muscle fiber model, we can see that little shallow depression on this model is our motor end plate. That's where you find chemically gated channels, and you also see that's where the ends of these motor neurons go to. It wouldn't make any sense for this motor neuron to hit the sarcolemma here. There aren't any receptors for those neurotransmitters. There would be nowhere for the neurotransmitter to dock. So we see neurons always talking to the motor end plate of muscles because that's the only place where there are receptors for it. This is more important than anything that we're going to pull down and see on the slides in just a second. But I feel like I just wanted to show a couple of last slides since we're talking about muscle contraction and the use of ATP. I'll leave that up there, and I'm terrible about that, but let me finish my thought here. So we have acetylcholine docks to receptors on the motor end plate. It causes those chemically gated channels to open up. We see more sodium goes into the cell than potassium goes out. So what I was saying, number 6, what that causes is a depolarization. This depolarizes the motor end plate, what we call the end plate potential. So end plate potential depolarizes. Again, that's just a way of saying that sodium ions are moving into the cell. And if that is sufficient enough, if this reaches the sarcolemma, I can't spell. If this reaches the sarcolemma, then voltage-gated channels open up. I'll just remind you, we got voltage gates for both potassium and for sodium. So if those voltage gates for sodium and potassium both open up, I know we haven't really drawn this yet, but sodium and potassium are going to rush in through these chambers, through our little channels here. It's going to cause a depolarization wave to go all the way down into this T-tubule, shake those voltage-gated channels open in the sarcoplasmic reticulum, and release those calcium ions. Those are the steps that I'm about to write on here. So end plate potential depolarizes the sarcolemma. Those voltage-gated channels open up. I'm going to say quickly here before I run out of space, in number 7, this wave of depolarization makes its way into the T-tubule and makes contact with the sarcoplasmic reticulum. We'll say enters T-tubule. And contacts that sarcoplasmic reticulum. So number 8, we're going to see calcium ions released from sarcoplasmic reticulum. If calcium ions are released from that sarcoplasmic reticulum, this is going to lead to muscle contraction. The opposite is true. If we want to get that muscle to relax, we just have to put away those calcium ions. And we said there's a calcium pump that's located on the surface of that sarcoplasmic reticulum that, like a vacuum, sucks out all these calcium ions from the sarcoplasm, stuffs them back into that sarcoplasmic reticulum, and what you're left with is a relaxed muscle.
Is it always going to be calcium, potassium, and sodium, or that's just an example that you use?
For the skeletal muscles, that will be the case.
Thank you.
No problem. When we get to neurons, talking to other neurons, it can sometimes change. All right, I feel like I'm doing okay on time. I just want to pull down a couple of slides. Is it okay if I pull that down yet? Did everybody get a chance to take a picture of that? I won't erase it, so it'll stay there even after I pull the slides down. You can take a picture of it now if you want, though. I feel like we've already seen 2 exam questions right there. Maybe more than that. We've got a muscle. We had the sketch of the epimysium, paramysium, endomysium, muscle fiber. We had over here the myofibril, the thick and thin filaments, light and dark bands. And then the most detailed thing here is how the neuron talks to the muscle. We finally have introduced chemically gated channels versus voltage gated channels that we can see they're located on different spots. Here we're just talking about where they're located on a muscle cell. But we started to introduce neurons a little bit. And I'm not going to erase anything. I'm just going to pull down these slides. So here we have our neuron that's coming down. We can see this axon terminal. The axon terminal, like we said, is covered in these voltage gated channels that are specific to calcium ions. And these gates are not going to open until an action potential arrives at the axon terminal. So, as long as they're closed, no calcium ions are moving into the axon terminal. None of these neurotransmitters are getting released. But this blue arrow is showing an action potential arriving at this axon terminal. And we know the first thing that action potential is going to do is it's going to open up these voltage-gated calcium channels. So that's the second thing that occurs. And then you can see as calcium moves into the cell, that's going to trigger the release of the neurotransmitter. So neurotransmitter isn't released until calcium ions move into the cell. We described kind of what was going on there. These are lined in a negative charge. The membrane itself is lined in a negative charge. So those repel one another until calcium comes in. It surrounds these vessels. Those positive charges are then drawn to the membrane's edge, and that leads to a release of this neurotransmitter. So neurotransmitter is released into the synaptic cleft. Number 4, we can see the diffusion of those neurotransmitters across that little space. And then finally, those neurotransmitters dock to those chemically gated receptors on the motor end plate. Notice that once we get out here to the sarcolemma, it's voltage-gated channels, but only down here in this little pit, this motor end plate, do we find these chemically gated channels. Then again, they open and close. If there are chemicals present, then this channel opens. If those chemicals are removed, then the channel closes. So chemically gated channel is what we're talking about. And then, like we said, chemically gated channels are also, they operate as non-specific ion channels. So when they're open, you can see it doesn't care what ion is crossing through it. It doesn't care whether it's sodium or potassium. It lets both of them move through. Now, for reasons that we're going to have to talk about later on, we're going to see that sodium comes into the cell more rapidly than potassium leaves. There's a reason, and I'll explain later. But what we need to know, since more sodium moves into the cell than potassium moves out, we have this wave of positive charges, this wave of depolarization that starts to spread along this motor end plate. And if it's sufficient enough, you can see the end plate, this end plate potential. If it's sufficiently disturbed, then we can see those voltage-gated channels are triggered. And if those are triggered, then it's a non-stop domino effect of an electrical signal going all the way down the sarcolemma and into the T-tubules the rest of once calcium ions are released, we can contract those myofibrils. What else is shown here that I didn't put in my diagram is that each one of these receptors has a little enzyme right there. This is an enzyme that breaks down neurotransmitters. In our example, this neurotransmitter was acetylcholine. Even here they're using the term acetylcholine as our example of the neurotransmitter. And these enzymes break down those neurotransmitters. So the blue structure there is representing this enzyme called acetylcholine esterase. It just breaks down acetylcholine. These neurotransmitters get broken down and taken right back up by this axon terminal, and they're recycled to make more neurotransmitter. Anyway, acetylcholine esterase. Well, you can see when acetylcholine esterase removes those chemical signals, the gate closes. So again, if the chemical gate is going to be open, there have to be sufficient chemicals present. So we've talked about chemically gated channels versus voltage gated channels. Here it shows when those when the end plate potential has been sufficiently disturbed that we've now triggered these voltage gated channels. This is when we first see our action potential. Again, action potentials are always going to involve voltage gated channels. When we're talking about chemically gated channels, it's a graded potential. Because you can imagine, if there weren't sufficient amounts of chemical signals, then this wave of depolarization would never get out of the motor end plate. We would never trigger those voltage gated channels. The muscle would never contract. It would be like me just lightly tapping on my knee over there. But if the signal is sufficient and that voltage gated channel makes it all the way down into the T-tubule, here we can see those voltage gated channels that really connect the T-tubule to that sarcoplasmic reticulum. And when that voltage gated channel, again, opens these calcium gates, calcium floods into this sarcoplasmic, floods into the sarcoplasm, the fluid of this muscle cell, and leads to the muscle contracting. We saw this last time. Active sites are covered up until calcium is on the scene, and then troponin prefers to hold on to calcium, and it lets go of those actin active sites. So now the active sites are exposed, and as soon as those active sites are exposed, we have this cross bridge that forms between actin and myosin. One of the last questions I had in there, which I think we've been talking about ATP a couple of times today. So in muscle contraction, ATP is actually going to be used to break that cross bridge once it forms. To be specific, there's two things that ATP is doing. The first thing that it does, we're going to see after the cross bridge forms, the only way we can break that cross bridge is by using one of those ATP molecules. The first thing that it does is it detaches actin and myosin, and the second thing that ATP molecule does is it cocks that myosin head back, just like the hammer on a revolver that you have to cock back. That's the whole thing in just a smaller series of pictures. I'm talking about the muscle contracting or relaxing. We know the role of calcium in exposing those active sites and getting the cross bridge to form. In thinking about the cross bridge cycle, there is one slide that I'm trying to get to, and it's this one right here. There's two ways of seeing it. We got that slide and this slide. They're both showing the same thing. And this is almost to the end of our PowerPoint, so I believe I'll make my 4 o'clock deadline. But we start right up here, which is right after calcium has arrived on the scene. Those active sites have been exposed on the actin, and immediately we get this cross bridge that forms between actin and myosin. I talked about that myosin head being like a hammer that's cocked back. And as soon as it forms the cross bridge, so we have actin and myosin that actually touch, the first thing that happens is that hammer is fired. That is what's described as the power stroke. And that hammer firing is what really drags those two Z lines closer together. We'll see this in a little animation. If it's not today, we'll see it as a review. But we did see that one animation where those two Z discs were pulled closer together. What's pulling those Z discs closer together, like we said, is these cross bridges that form between actin and myosin, and then that myosin does this power stroke to pull that actin rope along. Once the power stroke has occurred and this actin has been pulled along its journey, that cross bridge remains. And we said the only way to get that cross bridge to break apart is by using up an ATP molecule. Here's, like we were talking about a second, that it's currently an intact ATP molecule, but after we've used it to detach this myosin head and then cock that hammer back, that energy has been depleted. So it's now been kind of converted back to this dead battery, that ADP and the extra phosphate that's hanging off. We've extracted the energy from that. That what was once an ATP has now been broken down into an ADP. And we used that energy to break the cross bridge and cock that hammer back. So again, there's that ATP being used to break the cross bridge and cock this hammer back. So now we're ready for another cross bridge to form. This next slide, it shows the same thing, right? We've got a myosin head that's cocked back, and you can see that the ATP has already been ripped apart. Here is before we cock the hammer back. Like we said, we have to use that ATP molecule. We destroy that ATP molecule in cocking this myosin head back to the loaded position. So like we said, if there is no calcium ion present, then this myosin head stays in its locked and loaded position. When calcium ions show up, that allows this cross bridge to form between actin and myosin. We said as soon as that cross bridge forms, this myosin head does its power stroke. It pulls that actin molecule along its journey. So again, after that power stroke, you still have that connection between actin and myosin. The only way to break that cross bridge is to use another ATP molecule. The two things that ATP does, we said it detaches the connection between actin and myosin. That's the first thing it does. They were attached here. Now that ATP has detached the actin and myosin. The second thing that ATP does is cock back the myosin head. Down here, this is showing somebody that this is after death. Your body is no longer producing ATP. So if you're not producing ATP, there isn't any available ATP to break this cross bridge between actin and myosin. This is often what is the cause of rigor mortis in a dead body. If you feel those muscles that have stiffened up, that's due to the fact that there's no longer any available ATP to break these cross bridges between actin and myosin. And you have these muscles that are contracted, the slight constant contraction, at least for a few weeks until deterioration of those proteins occurs, and then the muscles finally relax. There are a couple of videos there we can see as an animation. And that is the last couple of slides. You can see we've seen this one already that we just used to show that these electrical events are always going to precede mechanical events of contraction and relaxation. The latent period here was due to the fact that it took a few milliseconds after this electrical event occurred for those calcium ions to actually show up in the sarcoplasm. Again, what's happening in those milliseconds is this electrical current is making its way out of the motor end plate, down the sarcolemma and down the T-tubule, hitting that sarcoplasmic reticulum, and then eventually we get calcium ions leaking into that sarcoplasm. As calcium ions increase, so does the strength of contraction. And as those calcium ions decrease, so does the strength of contraction.